Yo, 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 welcome to increasing genetic variation. And the big example here would be viruses. So, mutations, as we already know, are the ultimate source of genetic variation and create genetic variation in all life on Earth, both prokaryotic and eukaryotic. Now, there are many other processes that can allow organisms and species to acquire new combinations of DNA, leading to new genetic variation, which is important to life here on Earth. So, in eukaryotes, sexual reproduction, again, the process in to create the gamete, such as segregation of the chromosomes, independent assortment of the chromosomes, crossing over of homologous chromosomes, and finally the fertilization of or between two gametes. In prokaryotes, horizontal gene transfer, which includes the process of transformation, transduction, conjugation, and transposition. And we'll go over all of that. And finally, in viruses, recombination between related viruses is possible. Genetic variation in eukaryotes. So we have learned that the formation of the gametes involve these processes. Okay, and so the different combinations of the DNA in which a gamete is created again provides new genetic variations when those gametes are fertilized. And this is when a random egg cell is fertilized by a random sperm cell and it results in a zygote, as you guys can see here. And this zygote has new combination of DNA that has never existed before. Since prokaryotes reproduce asexually, again, the offspring are genetic replicas of the parents. But there is a way for these prokaryotes to create new genetic variations, and that's a process of horizontal gene transfer. So this process can be done during their life. Okay? Now, this is when an organism acquires genetic material from another organism that is relatively unrelated to, as opposed to a vertical gene transfer, which is the transmission of DNA from one parent to the offspring. So, as listed before, we have different processes of the horizontal gene transfer: transformation, transduction, conjugation. Let's start off with the first one, transformation. It occurs when a bacteria cell uptakes naked DNA, typically fragments of the DNA or plasmids from a surrounding, usually from a nearby lysed cell. So we can look at the image for the example there. The image shows us fragments of DNA, and then we have the chromosomal DNA of the bacteria here, and then it uptakes that AB DNA. Well, guess what? They degraded DNA again can break apart and it can attach to the chromosomal DNA. So the recombination occurs between donor DNA and the recipient DNA. And then the rest of it is just junk. As previously mentioned before, this is the second process, transduction. This is when foreign bacterial DNA is introduced into new bacteria cell by a bacteriophage, and it's summarized in this image here. So let's break it down. When a bacteriophage infects the DNA into a bacteria, it forces the cell to make copies of its genetic material and protein code. And then the viral DNA is loaded into the viral protein codes and are released from the cell in a normal process, right? Well, during the process of a virus attack on a bacteria, there are enzymes that are released to break up the DNA into fragments. Okay, so the bacteria cannot turn on its defensive genes. These bacterial DNA fragments can sometimes be loaded into the viral coats that are released by the cell in this step. So again, you can see that bacterial DNA lodged into the bacteriophage there. And so when those bacteriophages go to attack another bacteria, it injects the bacteria's DNA, and that DNA gets incorporated into the like the transduced cells chromosome. We see that fragment of the host DNA from this step here located on this bacteria's chromosome. The third process is called conjugation. This is when DNA, again, most commonly a plasmid, is transmitted between bacterial cells through direct contact. So the ability to do this is coming from the connecting bridge structure that is formed between the two bacteria called a pilus. So transformation and transduction does not require direct contact. This process, conjugation, requires this direct contact. The fourth one is transposition. So transposition occurs when segments in a DNA molecule relocate to different positions in the same molecule or relocate to a different DNA molecule. Okay, so in bacterial cells, this commonly occurs between plasmids and the bacterial chromosome, allowing certain genes from the chromosomes to become part of the plasmid and vice versa. And you guys can see that there. This part of the bacterial chromosome is then transposed into this plasmid. And plasmids are usually the ones that are being taken up by other bacteria. So it could either go through and get passed on by conjugation or transformation. There's a huge variety of viruses that exist. So we can see that over here. They're spherical, complex, helical, polyhedral. Some have the capsid, you know, envelope. They have the glycoproteins here. Many different varieties. There's a tail sheath, a tail fiber, capsomere of the capsid, but they are broadly categorized by the type of genetic material they carry. So for DNA viruses, they can either carry single-stranded DNA or double-stranded DNA. Same thing with RNA viruses. They could either carry single-stranded RNA or double-stranded RNA. So let's go over some of the important concepts of viruses. And this is dealing with bacteriophages. So the life cycle of bacteriophages has two parts, the lytic and then the lysogenic. So with the lytic cycle, we saw that with that transduction process, right? But in transduction, the fragmented bacterial DNA is incorporated into one of the bacteriophages and then transferred to another bacteria. However, this is just the first part of it, where the bacteriophage, the virus, injects this DNA, uses the RNA polymerase and then the ribosomes to produce the proteins and parts of the virus. They assemble within the bacteria, and when there's just too many of the viruses, the bacteria cell lyse, releasing all of the bacteriophages. So the lytic cycle is considered a very quick cycle in terms of no incubation period. So it causes the bacteria to lyse, thus lytic. Okay. The other part of the bacteriophage's life cycle would be the lysogenic cycle. The lysogenic cycle replicates the phage genome without destroying the host. So it incorporates that DNA into the bacterial chromosomal DNA, and it just sits there getting replicated over and over again when that bacteria undergoes asexual reproduction. And so the lysogenic cycle just repeats itself in this process until the lysogenic can turn into lytic, as you guys can see in this diagram. The biggest thing with viruses for the AP exam is the concept of the retrovirus. So what is a retrovirus? First of all, let's get this straight. It is an RNA virus that inserts a copy of its genome into the DNA. And so, to accomplish this, the virus's RNA must be converted into DNA. That's the key. And there is one enzyme that the retrovirus contains that's called reverse transcriptase in order to do so. Okay, so the DNA can then be integrated into the host cell's chromosomes and will ultimately be transcribed and translated for the viral progeny. So let's take a look at the process. Viral RNA. Oh no, reverse transcriptase converts that RNA into DNA, back to RNA, to the proteins. Okay, so there we have it. That is the flow of genetic information for retroviruses. It goes RNA to DNA, and then that DNA gets transcribed, then translated into the protein parts. Well, can't stress this concept enough, so you're seeing it one more time. We have HIV. With the glycoproteins allows the HIV to enter and release its content to the host cell. Within the contents, y'all, we see the viral capsid proteins, as well as reverse transcriptase and RNA, since HIV is a retrovirus. So from there, the retrovirus RNA gets reverse transcribed and it gets turned back to DNA. There is double-stranded, and so it enters the nucleus and gets incorporated into the chromosomal DNA of the eukaryote. Now, mRNA can be created from this, and its parts are created. So we have the RNA genome for the virus being created. We have the mRNA here being translated into the parts of the virus, and therefore it can create the different parts and again send it to create a new HIV virus within, the area. So it just hijacks the system, y'all, and creates more HIV retroviruses. So viruses evolve quickly, often faster than their hosts, but how? So first off, viruses, just like cells, can acquire mutations. And just like other cells, a large majority of these mutations occur from mistakes that are made when their genomes are replicated inside host cells. The DNA viruses, y'all, are replicated using the cell's DNA polymerase enzymes, which have proofreading function. But RNA viruses must use their own special enzymes, RNA-dependent RNA polymerases, to make copies of their genetic material. However, these polymerases do not have a proofreading function and they end up making more mistakes, and so RNA viruses generally mutate much faster than DNA viruses. So again, why are they evolving much quicker? Viruses, right? So viruses reproduce very quickly with high mutation rates, as seen on the previous slide, and many genetic variants can arise just from the mutations alone. The last reason, again, is that viruses can also undergo recombination, a process that happens when similar viruses infect the same cell and their genetic material ends up packaged together, making a new virus. And this, again, is usually the basis for the influenza virus, the flu, right? Again, many different strains because of this process of recombination. Okay, y'all, that's it for today. Have a good one.